Hello everyone, welcome back to Online Learning with Miss E. This is the Central Dogma of Molecular Biology video part 3. So if you have not seen parts 1 and 2, make sure to watch those first so this one will make a lot more sense. But today in part 3, we are going to talk about translation, going from RNA to proteins. Before I jump into the content, in case anyone happened to be wondering what Miss E has been up to, here's a picture of a 1,000 piece puzzle that my roommate and I made last week while we were social distancing. During this time, I've also been taking online classes just like you guys for graduate school, practiced my Spanish, I've been playing soccer, and I've been going on a lot of hikes, foraging for mushrooms, because it's morel mushroom season. So that's what I've been up to. Let me know what you guys have been up to. Comment in the Google Classroom. I'd love to know what you guys are doing. But for now, let's jump into the content. Let's talk about translation. So, a quick recap from the last two videos. In video part 1, we talked about the structure of DNA. In part 2, we talked about the first step of the central dogma. We talked about transcription, going from DNA to our RNA. And today we're going to ask the question, how does RNA facilitate the production of proteins? So that second step of the central dogma, how do we go from RNA to proteins? So the way we go from RNA to proteins is through the process of translation. Translation is just the decoding of an mRNA message into a polypeptide chain or a protein. You can see in this picture here, it kind of summarizes the whole central dogma. First, we go from DNA to RNA and transcription. We transcribe that DNA. And then in translation, what we talk about today, we're going to go from mRNA to protein by decoding this mRNA message to figure out what amino acids we have to put together to make a protein. Because remember, amino acids are the building blocks of proteins. All right, so we mentioned decoding the mRNA. We can think of RNA as a genetic code, as a code that we have to crack. So the sequence of bases in our RNA will tell us what amino acids to put in order to make the protein that we want. And this genetic code, the mRNA, is going to be read 3 letters at a time, 3 nitrogen bases at a time, so that each word of the coded message is 3 bases long. And each 3-letter word in mRNA is known as a codon, and it codes for 1 amino acid. So if we look in this picture, each codon, we have 3 bases, 1, 2, 3. And so that's going to be like 1 word, and it's going to code for 1 amino acid. So in this picture, we have 2 codons, so we would have 2 amino acids. All right, so let's look at an example. So imagine that we had this mRNA strand, and these are nitrogen bases, UCG, CAC, GGU. How would we read this as codons? How would we read this code to make our protein? Well, we would go 3 bases at a time. So our first codon would be UCG. And then we would have CAC. And then our last 3-letter codon, 3-letter word, would be GGU. So how many amino acids would this code for? 3. Each codon, 1, 2, 3, codes for 1 amino acid. In this case, our amino acids would be serine, histidine, and glycine. But you guys don't need to know those specifics right yet. Okay, so again, look at this picture over here. If these are nitrogen bases, you would read them 3 at a time to get your 3-letter codon, which ultimately codes for an amino acid. All right, so let's do some practice. If this is an mRNA strand with nitrogen bases, how many codons are shown here? Well, each codon is going to be 3 bases. So I'd have 1, 2, 3, 4, and 5. So I would have 5 codons. which means I would have how many amino acids? Yep, 5. 5 codons, so we would have 5 amino acids here. A little bit more about the genetic code, because there are 4 different bases. Remember, those are adenine, guanine, cytosine, and uracil for RNA. There are going to be 64 possible 3-base codons. And so that's a lot of different possible codons, and there's a lot of different amino acids that they could code for. So we have an amino acid chart, which you can see here, that will show all the possible 3-base codons and the amino acid that they code for. So this is super helpful for us to be able to figure out which amino acids we have. So for example, if I gave you a codon that was CUG, and I asked you, what amino acid does that code for? We can use this chart. So we start in the middle. The first letter, the first nitrogen base in this codon is a C. So I would find my C here. So I'm now in this corner of the grid. Okay? Then we go to our second letter. It's U. So I follow it along from C to U. And then my last letter is a G. And I can see that the G here codes for the amino acid leucine. Okay? Let's try one more. If I gave us AGU, if my codon was AGU, start in the middle from A, G, U, and I can see that our amino acid is going to be serine. And you might also notice in this amino acid chart that there are some codons that will either start protein synthesis or that can stop protein synthesis. So these codons are important because they either tell the protein to start being made or they tell us when to stop making a protein. It tells us when the protein is finished. So this amino acid chart will either tell us what amino acid should be brought in or to start or stop protein synthesis. Okay, so here's just another picture with some examples. Again, we have a codon here. It's GCU, our first 3-letter codon. So if I went GCU in the chart, you can see our amino acid is alanine. And then we have our second codon, ACG. If I use my amino acid chart, again, we start in the middle of the circle. I start with A, C, G. We get our amino acid threonine. Okay? That's how we use the amino acid chart. So now I want you guys to do some practice. Use the amino acid chart here to identify the following amino acids. As an example, did the first one for you, AUG. This is going to be our start codon. So again, start in the middle, A, follow down U, G, and we get that start codon. All right, so pause the video here. There are 3 more for you to try on your own. Give it a shot and then come back to me. All right, did you guys try them? Let's see. Our first is GCU. If I use this amino acid table, GCU, you should have gotten alanine, ALA. Our next one, CUA, CUA, should have brought you to leucine, LEU. And our last one, UAG. If I go UAG, that actually brings us to a stop codon. So that means that the protein has been made, we are done, and that protein is finished. Okay, did we get that? If not, I would go back, again, listen to the video talking about the amino acid chart again, and then give this another shot. All right. So how does translation actually work? Well, after mRNA is transcribed in the nucleus, we remember that it leaves the nucleus to enter the cytoplasm and will attach to a ribosome. And ribosomes are that organelle that are the site of translation. So you can see in the picture here, this big kind of blue-green guy, that's our ribosome. And the mRNA is going to attach to the ribosome, and translation will occur at the ribosome. It's like our factory that makes proteins. So let's talk a little more about that ribosome because it's really important. The ribosome is made up of two different subunits, so it's made of two parts. It has the large subunit, which is called 50S, and the small subunit, which is called 30S. And you can think of this like a hamburger bun. It has two sides, and they're going to clamp together, and that mRNA is going to be like, I don't know, your burger or your lettuce or whatever you want to put in between those two hamburger buns. Okay? All right, so these two units of the ribosome actually exist separately in the cytoplasm, but they will join together on the mRNA. They will clamp down on that mRNA. And the ribosomal subunits contain two types of RNA, rRNA and tRNA. Do we remember those? Do those sound familiar? Let's review the three types of RNA really quick. So firstly, our first type of RNA was that messenger RNA. It brings information from the DNA out to the cytoplasm. This is where we have our codons that we read 3 at a time. Next, we have our ribosomal RNA, which is the actual ribosome. It's our two hamburger buns. And those large and small subunits will clamp onto the mRNA and read it to assemble amino acids in the right order. And then the tRNA, which also goes along with the ribosomes, transfers the correct amino acids onto the ribosome. So it is our transfer RNA. And each tRNA has an anticodon. Let's look at this picture here. There's an anticodon whose bases are complementary to the codon on the mRNA. And that's how that transfer RNA can attach to the mRNA and bring the right amino acid. So if we look at this picture, this whole guy right here is our transfer RNA. The anticodon is on the bottom with the complementary nucleotides. So we have A with U with A, G with C. And when it bonds to the mRNA, it brings along the correct amino acid. Okay, so how does translation work? How do these three types of RNA work together? So translation begins at AUG. That is our start codon on the mRNA. So here we have mRNA. So we remember what type of RNA we're looking at. And you can see the start codon here, AUG. And this codon tells the ribosome to grab on and to start translating at this spot. All right, so the ribosome, you can see in this animation, attaches at the start codon, and the tRNA, our transfer RNA, will bring that first amino acid. So in this video, we see the ribosome clamping down, and our transfer RNA with its anticodon, its anticodon is right about here, that bonds to the mRNA and brings the amino acid that you see up here. All right, once the ribosome has attached and it's brought that first amino acid, the ribosome will keep reading the mRNA strand 3 bases or 1 codon at a time, and the tRNA with the complementary anticodon will just keep coming in and bringing the right amino acid, one after the other. And those amino acids will attach via peptide bonds. So, if we look at this picture here, we have our messenger RNA. It's this long green one. It's attached to our ribosome, and you can see your tRNAs are coming in. They have that matching anticodon to be complementary to our codons, and then all these amino acids are just being attached by peptide bonds. So that little line in the middle is our peptide bond. And we're slowly making a protein. So it's just going to keep reading the mRNA. The tRNA will keep bringing amino acids. Here's a little picture of that. So you can see your tRNA is coming in. The anticodon is attaching to the correct codon, and it's bringing in its amino acid and making peptide bonds to join those amino acids together. Just made a peptide bond there. Next tRNA is coming in. Another peptide bond. We're building our protein. All right, so when do we stop? The process is going to keep going until the ribosome reaches one of 3 stop codons. And at this stop codon, the completed protein will be released from the ribosome into the cytoplasm, where it will go wherever it has to go. So our 3 stop codons, you can see here, they are UAA, UAG, and UGA. And now, for right now, you don't need to memorize them, but a helpful mnemonic to remember if you ever do need to memorize these stop codons. UAA, you are annoying. UGA, you go away. UAG, you are gone. Not super friendly, but if they help you, give it a shot. Maybe it'll help you remember your stop codons when you have to remember those someday. Okay, so now let's just walk through another little virtual modeling of translation. Again, I'll post a link to this website if you want to try it on your own. But for now, let's take a look here. We have a little picture of some cells, and let's actually do a quick review of transcription. So we're entering the cell now. We're going into the nucleus. And inside the nucleus, we can see our double-stranded DNA with our sugar phosphate backbone, our bases on the inside. RNA polymerase has showed up. It is attached to the DNA, opened up that DNA, and we are now copying the template strand of our DNA, adding complementary bases. And you will notice that uracil is in place of thymines, and that RNA polymerase is just going to keep building our mRNA strand until it's done. Okay, now this mRNA strand is finished. It is ready to leave the nucleus out into the cytoplasm to facilitate the production of proteins. So now let's go to translation. Okay, we are in the cytoplasm now. You can see our mRNA here. The big brown ribosome has attached, and our transfer RNA has attached to the mRNA. You can see its anticodons here that are complementary to the mRNA strand, and it's brought our first amino acid. So now translation will continue. Our next tRNA will come in, and it'll bring the next amino acid, and you can see that the 2 amino acids have created that peptide bond. And we'll just bring in the next tRNA and lengthen that protein chain. And the mRNA is going to keep being read, again, 3 bases or 1 codon at a time. And so the codons will keep being read, telling us which amino acids to bring next. And we just keep bringing amino acid after amino acid. And you can see that protein's getting longer, it's starting to fold in on itself, and eventually we are going to reach our stop codon. That protein is released, and we're done. Okay, so if you want to try that on your own, again, the link will be attached underneath this video. But for now, that is the end of translation, and we have made a fully functioning protein. And so our central dogma of molecular biology is complete. We have gone from DNA to RNA in transcription, and then we have gone from RNA to proteins in translation. So we have gone from the DNA that we inherited in our genes to the actual proteins our body needs to function. We have gone from the traits we inherit to the expression of those traits, and that is the central dogma of molecular biology. Okay, so this is where we are now. DNA was transcribed into mRNA in the nucleus. The mRNA was shipped out to the cytoplasm and attached to a ribosome, and that ribosome translated the mRNA into proteins. To do that, it read the mRNA 3 bases at a time, and 3 bases, again, is 1 codon that codes for 1 amino acid. The start codon is going to be AUG, and then tRNA will continue adding the correct amino acids into a growing polypeptide chain until we've reached a stop codon, at which time the ribosome will come off the mRNA, that protein will be released. And that is it, guys. This is the end of part 3 of 3. You have completed the entire central dogma of biology. Congratulations. And as always, please post any questions or comments you have in the Google Classroom. We are here to help you work through this material. If there's anything that you're confused about or anything that you just thought, hey, this is really interesting, I would love to learn more, please let us know. We are here to help with any and all things. And also reach out to Mrs. Liu or I if there's anything we can do to support you. Otherwise, keep checking the Google Classroom for other activities. And congratulations, guys, you did it. All right, Miss E is signing off.